Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Clinical Trial Considerations in Cerebral Protection for TAVR Patients. My name is Andrew Jurinen, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, and that's on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using that chat panel. And at this time, all participants are in a listen-only mode, and please note that this event will be recorded and made available for future download. Now, I'd like to introduce our speakers for today's webinar. Dr. Alan Lumsden is the Medical Director for Medical Metrics and a faculty member at Houston Methodist. Dr. Lumsden has developed an international reputation as a leader in the field of endovascular surgery. He has clinical and research expertise in stent graft treatment of thoracic and abdominal aortic aneurysms, stenting and endarterectomy in carotid arterial disease, renovascular hypertension, aortoiliac occlusive disease, mesenteric vascular, and minimally invasive therapy in venous disease. Dr. Lumsden also conducts FDA-mandated training for surgeons nationwide in a carotid stenting simulator housed at the Houston Methodist DeBakey Heart and Vascular Center. And your second speaker for today, Dr. Zsolt Garami, is the Director of Vascular Imaging for Medical Metrics and a faculty member at Houston Methodist. Dr. Garami's research focuses on understanding and treating intracranial flow disturbances that cause stroke. His current studies aim to reveal critical patterns of distal intracranial embolization of LVAD in TAVR patients. And finally, Rochelle Massey provides strategic leadership and program management for Novella Clinical's Medical Device and Diagnostic Division. She has more than 11 years of medical device and clinical trial management experience, including clinical trial strategy for enrollment, execution, vendor management, and regulatory approval. Mrs. Massey has supported programs for a number of Class 3 type devices, including structural heart devices, drug-eluting stents, and bioreabsorbable scaffolds. And now, it's with my pleasure to hand the microphone and the presentation over to your first speaker for today, and that's Dr. Alan Lumsden.
All right, so first of all, welcome. And I'm going to talk and focus today about clinical trial considerations in cerebral protection for TAVR. Okay, so let me tell you a little bit about my background. First of all, I'm a vascular surgeon. I'm the chairman of the Department of Cardiovascular Surgery. And my main interest has been around stent grafting and increasingly around thoracic aortic stent grafting. And one of the major concerns and one of the major complications from this, of course, is stroke. As my role as the department chair, we also have a very robust TAVR program. We're one of the top enrolling sites in the United States, and one of my partners, Dr. Mike Reardon, is the national PI in both the CoreValve trial and the Lotus trial. And so we have a lot of experience. We work very closely together, and one of our areas of interest is in stroke and stroke prevention. Dr. Garami is really one of the world's experts in transcranial Doppler, and we brought him into our department specifically because we fundamentally believe that TCD can give us a very interesting analytical insight into when and where embolic events occur during these procedures. So, next slide, please. So, what we're going to talk about is the rationale for cerebral embolic protection. And TAVR, of course, has really exploded in the utilization both clinical application now and also in research trials. And it has become a very useful alternative to surgical valve replacement. Now, As with many trials, of course, these start off as being applied only in a high-risk population. But as you'll see in subsequent slides, that what we're looking at is the motion is to lower risk and broadening out the number of patients. The recent meta-analysis showed that clinical stroke rates versus TAVR were somewhat similar when you looked at them at 30 days in high-risk patients, but with an increased risk of microinfarcts. And this is going to be a lot of the focus of this talk, is what is the importance of microinfarcts or neuroimaging demonstrated lesions inside the brain. So if you look at that table, the procedure, surgical aortic valve, the clinical reported stroke rate is in the order of about 0.5 to 62%. Now, even if we were looking at coronary bypass, it's down in the 0.5 to 1.5% range. So just intervening on the ascending aorta, whether you're putting a valve in or whether you're doing a bypass graft, is associated with a risk of clinical stroke. Indeed, any intervention that involves crossing the aortic arch, even something as relatively simple as coronary bypass, cardiac catheterization, is associated with some risk. In TAVR, you can see the clinical stroke rate was 0 to 4.9%. The original Edwards trial was much higher than this at the moment. So companies have done a good job in reducing the profile, but if we look at microinfarcts, we're going to show you that this is still a significant problem. So silent stroke identified on diffusion-weighted MR still occurs in the 68 to 93% range. Only a minority of the patients with these cerebral lesions will develop a neurological deficit, and that has been the source of great controversy about what the ultimate importance of this. I'm going to advance the slide. So what is stroke? Again, the clinical manifestations of stroke may vary. They may be first of all, you may have a lesion in the brain which has no clinical manifestation, at least not that we can pick up. Now, we know that the more diligent you are, for example, if you have a group of cardiologists and CV surgeons evaluating the patient, we will not pick up as many clinically manifestations of stroke as, for example, a stroke neurologist. So to some extent, it's about how detailed that clinical exam actually is. The revised American Heart ASA guidelines in 2013 started to raise this concept and define stroke as any objective evidence of neurological infarction, including MRI findings. And it's this that's really started to get, certainly got my attention as this has been brought up to me. In the more recent publication by Lansky from the NeuroArt group, suggesting that microinfarction and neuroimaging is now considered stroke, is something that really got my attention, and we'll talk a little bit more about that. So this was the NeuroArt. This is the neurological group from the Academic Research Consortium, and if you look at the authors, this is a multidisciplinary group of pretty high-powered people, including cardiologists from the TAVR world and neurologists, all of whom are coming together to try and propose a standardized neurological endpoints for cardiovascular clinical trials. And so this is something that I think we need to look at in a little bit more detail. So we move on to the NeuroArt definition of stroke, is that they define stroke now, and that's the key word here, is talking about the definition of stroke. Type 1 is kind of what we've all regarded as clinically evident stroke. Patient has an infarct, they have a symptomatic stroke that we can detect clinically. They now define type 2 stroke. Now again, when we now think about how this conversation is going to go with a patient, the definition of stroke and trying to explain some of the nuances of this to patients may be somewhat challenging. A type 2 stroke is defined as covert CNS injury, i.e. silent asymptomatic stroke. We find lesions, diffusion-weighted MRI positive lesions that are positive on neuroimaging. And we're going to show you just how prevalent this happens to be. Type 3 neurological dysfunction is essentially where there is no imaging to show the presence of stroke and no manifestations basically other than transient dysfunction. In other words, probably interruption of the function of the neurons, but no anatomical abnormality, and patients recover over that. So we're going to go on now to the next slide and look at post-TAVR clinical stroke rate. That would be type 1 stroke. And you can see that it's variable. Some report from Walter was down at 0, but others basically have been as high as 5.5%. The most recent study, and this is my partner, Mike Reardon, this was presented at ACC and was synchronously published in the New England Journal, shows there was about a 3.4% stroke rate. Now, Obviously, that is not terrible, but even a 3% stroke rate carries significant morbidity and societal cost in terms of managing these patients. Those are just the patients who are being picked up on neurological examination, and this was the first trial that was being reported out on an intermediate risk group of patients. It was the biggest one on an intermediate risk group of patients. So these are not even the high risk or extreme risk group of patients who you would expect it to be a little higher on. In other words, it doesn't seem to be improving that much just based upon the acuity of the patient selection. So one of the things we've been interested in is, when does stroke occur? And so this is where I'm going to try and persuade you of some of the importance to us of monitoring. Whether the patient has a TAVR implanted, whether they're asleep or they're awake, even when they're awake, they're sedated. And so it's often very difficult, if you just purely go on clinical manifestations, to know when an event actually occurred. So let's say, for example, the patient is asleep, they're waking up, you evaluate them, they've got a stroke. Well, then the question is, when did that actually occur? And so we're going to move from looking at hour to hour and day to day to what we think is potentially minute to minute sensitivity in determining when a stroke likely occurred. So when does a stroke occur? 95% of these strokes are ischemic. That is to differentiate them from intracranial hemorrhage. So that's what we'd expect. These are particles, most likely, which are going on up into the brain and lodging in some of these vessels. The median time to major stroke, clinical stroke, is 1 day. That probably means it occurred in and around the procedure. And cerebrovascular events will most frequently occur in days 0 to 1, i.e. that is procedure related, and over half of these are major strokes. So that is a very significant finding. And you can see really these multi-center cohorts are just charts out what the timing of these strokes happen to be. Now, it would be very nice if you could break down the procedure itself and have some idea when these embolic events are actually occurring during the procedure. So let's take a step back, go on to the next slide, post-TAVR brain lesions and MRI. So if we start looking for these lesions, and a lot of people don't want to look because they're so prevalent, then we've known that this is fairly common. And if the green bar really is a polled estimate that about almost 80% of people will have positive lesions on MR. And our specialty has largely dismissed this. And I've kind of dismissed it because we couldn't see a clinical manifestation and really didn't know what was going to happen with these patients long term. Well, according to this NeuroArc classification, that is a stroke. And the question is, are we going to now have to talk to our patients preoperatively and say, well, 80% risk you're going to have a stroke. The majority of them are not really that important, but it's defined as neuronal necrosis. And it's estimated that about 1 cubic millimeter of positive diffusion-weighted MRI represents about 80,000 dead neurons, and on average, these patients have 5 to 7 cubic millimeters. So you can do the math and see that that's a challenging conversation we're going to have with these patients if we're going to live with this classification system. So let's move on and talk about the role then of embolic protection. And one of the earliest devices actually was the umbrella. It was actually developed by a vascular surgeon out of UPenn, and the whole idea was it came in through the arm of the umbrella and it deflected the particles away from the supraortic trunks. It didn't cover the left subclavian, so it wasn't completely protective. The whole idea was to prevent those particles going on up into the brain. And what are those particles made of? Well, the short answer is they can be made of a variety of different things. It may be calcified plaque, it may be pieces of the arterial wall, it could be thrombus, which we actually knock off. And remember, the way that devices are navigated to the aortic valve or to the corneostia, navigation is not what happens. Basically, we depend upon a series of wall interactions between the device and the outer wall of the aorta to have that device move from the femoral artery over the aortic arch to the target. And so when we as surgeons look inside that aortic arch, we're kind of surprised that it doesn't happen more often than it actually seems to do, because these, and the patients that we're treating, they are lined with pieces of thrombus, organized thrombus, calcification, irregular plaque. So that's kind of what gets knocked off. And then when you actually inflate a valvuloplasty balloon or the device in the aortic valve, then you further add to that. And so it's generally estimated, and Dr. Graham will talk a little bit more about this, is that debris more than about 250 to 40 microns can cause stroke. And if you look in some of these capture devices versus deflection devices, then the majority of these particles are greater than about 500 microns. So they look like they're likely pretty significant. Now, my interest in this, basically, really, this is a paper we published in the Journal of Vascular Surgery, and it was because of our concern about when did embolization occur as we were doing procedures in the aortic arch. And so we use transcranial Doppler because it will tell us when embolization is occurring. And so it's this link to what I, as the interventionalist, did and what is going on up in the head real time that we think is incredibly important. Now, one of the often criticisms of transcranial Doppler monitoring is it's very sensitive. It'll detect particles that are less than 250 microns. And so a lot of people say there's a lot of noise. What I would turn that around and say, when you see no embolization by transcranial Doppler, then I believe you can take that to the bank, that the event did not occur during that phase of absent embolization. And so it allows us to help refine the procedure by identifying what the at-risk maneuvers are. So, for example, in thoracic stent grafting, it was deploying a stent graft itself and then post-deployment dilation of the balloon that was associated with embolization. And so it allows you to very precisely tune the procedure and give feedback to the engineers about what is most likely the most embologenic period of this. So if we move on now and talk a little bit about the different devices, I mean, essentially there are capture devices such as Sentinel, and there are deflection devices you can see in TriGuard. They're all basically delivered through relatively small delivery systems, but there's a big difference in terms basically of the materials and the porosity. And occasionally you can actually find these can be thrombogenic in their own right, and how do we actually manage this? And so we're in a world where there is concern about the amount of clinical stroke. There is real concern that if we're going to be held to a standard of reporting type 2 strokes and trying to evaluate the incidence of that because there's concern about it does have clinical consequence, then fine-tuning the positioning or the composition of these filters may be very important. And if you look at the next slide on the current technology and clinical status, you can see that some of these are already being used in Europe, and some of them basically first in man as planned, and others basically are waiting on 510 clearance in a variety of different scenarios. So that's a little bit of the background about why I got interested in this, why our Department of Cardiovascular Surgery is extremely interested in mitigating the risk of stroke, and how the recent reclassification of stroke has just heightened our concern about how this is going to be explained to the patient. And, with the new embolization prevention devices coming online, how do we actually judge clinically, real time, and both with imaging after the fact to try and identify which patients are at risk and how we mitigate that. And so with that, I'm going to hand this over to Dr. Garami.
Hello, everybody. I would like to continue with stroke imaging, and I would like to talk about the MRI first, how we are using the MRI technology. The diffusion-weighted image is our gold standard for the acute stroke imaging and microinfarct. And basically, what it makes it really special that we're able to detect any extracellular free water molecule motions, and basically when the cell dies and we have a swollen cell due to the edema, is going to restrict the extracellular space. Basically in the brain we have 80% intracellular and probably about 20% extracellular water, and in a moment this water motion is restricted, we're going to have this beautiful bright hyper-intense signal on a DWI scan. The DWI scan is really fast, so it's really free of patient motion artifact, and it's really positive within seconds to minutes after a recent acute stroke. This what makes the DWI really the most reliable method for the early detection of the cerebral ischemia. And this helps us to, again, differentiate from the ischemic stroke from other disease process that could mimic stroke. It's very interesting that you can see in this picture what's the importance of the baseline DWI. The baseline DWI here shows no lesion, and it's very important because some meta-analysis showed that at baseline we can have 32% atrial fibrillation already present, and that could be a major source of microembolization. And also we can encounter about 10% recent stroke. So the baseline DWI would be one of the exclusion criteria. The red arrow shows a lesion on the right side. So basically on an MRI, this is your right side, this is your left side. And these are the two lesions we can identify. With this hyperintense signal on the DWI, very important sequences are ADC. This is a apparent diffusion coefficient map, and it's actually a true value calculated from two DWI B values, the B0 and the B1000. And these values are able to help us and confirm that these lesions, what we see on a DWI, they are really present and they are true acute event or what we see, and they are not an old chronic process which could be the T2 shine through effect. So with these true values, what we can see is that with a normal CSF 3200, the normal white matters on 800, the core of these ischemic lesions running around 400, and these values are in millimeter square per second. The important is to really go through the standardized MRI protocol so we can really facilitate the comparison through across the study, and with the DWI and ADC map, we definitely would love to have a picture of the old lesions. And the flare, this is our fluid attenuation inversion recovery picture, which looks like a T2, but the water now is dark, and able to differentiate that when you had these two lesions from a previous slide, then you can see the dark on the ADC map, and then suddenly you still don't see it on the flare. So it takes time to show up on the flare, but what you do see is all the previous stroke would happen bilaterally in both hemispheres, and again these are accumulative throughout the past, and again this is just a snapshot. While you're looking at the DWI, this is positive again only up to 7 days. So after 7 days, these positive findings may be gone and going to observe on a T2 and a flare pictures. The other important frequency and sequences what we are trying to use is one of the magnetization prepared rapid echo. This is the MP-RAGE, and also the spoiled gradient echo. These are basically allowing us to do a 3D reconstructions, and all those 3D reconstructions we also can do some post-processing that any distortion we may see we're able to make some corrections. So these geometric corrections are important because sometimes around the sinuses and the mastoid cells we may have problem defining the lesions, so it's not a problem when we're just counting them. But if you want to find a quantitative volumetric analysis, this becomes important. So these are most often will be done in the core lab and not at the site, and these are allowing us to able to co-register and also generate some substruction maps similar that these dual echo are used for the B0 correction in the clean TAVR trial as well. Another very important thing is, again, we started to talk about how long the DWI remains positive. This is our own publication from Dr. Fang, And what it shows, again, 6, 9, 30 hours and 5 days and 3 months. So it's very interesting that in the previous studies, when we see the 3 months DWI, 80% disappears, and at 6 months, all of them disappears. And you can see the brightest DWI lesion you're going to see about 30 hours after the stroke. You can see the hypodensity on the ADC map, and I think you can really appreciate it, how easy to really confirm the acute lesion on the ADC map. So this is bringing us again the timeline where we would like to do the post DWI. We would like to time it to 24-48 after the procedure. Basically, if you have a wide window of 2 to 7 days, it will encounter like heterogeneity of the whole data, and the heterogeneity would be minimized if you really narrow this window down. When we do a APD trial, we really want to see that 2 hours procedure really what's responsible for those emboli. We are not proposing a TAVR trial when you really want to see the whole valve responsible for up to 30 days stroke rate. So I think the follow-up is really optional, and previous trials showed that these DWI lesion size becomes smaller, almost like cut in half. So if you do a day 2 DWI, by day 7 the size of it becomes almost like half. The other important thing is also that the lesions, how we can measure in the certain areas, and I think the location we'll talk about in the next slide in a moment. On this slide, we wanted to share with you all the prospective studies with EPD control groups published, and these are the trials with a number of patients, and I wanted to point out that on the recent trials, all the imaging use the 3T MR technique, and you can see that few of them use the MR together with the TCD. And I think it's very important because the signal to noise ratio is directly proportional to, again, the magnetic field, but also this determines the quality of the image. Also on the 3T, we can do faster imaging, and the only problem we may encounter that we may have more artifact on the 3T, but I think we can do some correction on those. The only limitation again for 3T is the pacemaker, so the contraindication for pacemaker we can again allow to do a 1.5 Tesla MRI used. And as you can see that on the follow-up MRI, when you have a wide open protocol, we really would like to recommend to follow the clean TAVI, and again, leaving the day 7 as optional, but really focus that the follow-up MRI should be done 24-48 hours, definitely after the procedure. Also, because the size of the lesions are very small in these TAVR procedures, we need to really utilize multiple orientation on the DWI, so we can, So what we're trying to do is that with the axial, coronal, and sagittal planes, we're able to visualize in multiple views. We can give a different color for the different lesions, and that able to track them in a 3D environment. So I think the size, the number, and also the volume at the given location will be nicely tracked in this 3D MRI environment. And as you may know, the importance of the whole brain perfusion, if we count 100%, done by the 40% by the 2 carotid and 10% by the 2 vertebrals. So for that reason, we would like to offer a 4 region as anterior cerebral artery, middle cerebral artery, posterior cerebral artery, and the vertebrobasilar system as again for the location. And I think it's unnecessary to have like 28 different brain territories as used in the Sentinel study because I think the 4 main region is really appropriate for the supplying large vessels. And by mapping our lesions from the previous case, you can see that we have MCA related, and then we have PCA related flow disturbances, and these are the DWI lesions tracked and colored, and this way we can also measure it in multiple cuts. One more important thing is that you need to get ready for some anatomic variation when a PCA may come from the carotid. The fetal origin PC is the most frequent again anatomic variation, and that's why I think it's good to have some brain imaging done. This table really shows from Pagnesi a nice meta-analysis that shows that the single lesion and also the total lesion volume really shows significant difference between the EPD and a non-EPD group. What we did not find any statistically significant difference here with a new lesion numbers. But I think it's really surprising that the wide range of this lesion, and it's very important that the size of about 29 cc is not going to reverse, and this is where we have definite positive DWI lesion which remains for 7 days. So for as a clinical site, we definitely will propose a strict protocol, but also in the site we'll test and select the equipment for the proper for the study, testing with the Phantom, and again in the core lab facility we'll definitely do the post-processing and any additional work needed. And finally, I would like to talk about my TCD experience and also some TCD data available. And I think the TCD with the real-time tracking is really can give us, as Dr. Lumsden mentioned, a clue during the procedure what's going on, why MRI is just giving you an accumulative end results for everything what happened. And again, TCD does more than just counting emboli. It's giving us some really nice hemodynamic information, for example, in the rapid pacing when the valve is positioned. The highest embolization, what we saw in publication in our own experience, is definitely the valve positioning and the valve deployment. But I think it's very important in these studies when we're comparing different valve devices and different methods that TCD can really define and make a difference when we are trying to stratify, again, if we have pre and post balloon or we have different technique for the valve implantation. Finally, I would like to finish with 2 examples. These are snapshots of video recording of TCD during a procedure. You can see that here is a Edwards sapient going to cross the arch. This is the TCD simultaneous second channel timestamp and simultaneously recorded. These signals are coming from the red blood cells. If anything else is traveling in a red blood cells, going to give us the high intensity transient signals. And these high intensity transient signals, surprisingly, they were quite rare. In the ProTAVI, it was like average of 630 and higher in the APD group than in the control group. But in the clean TAVI, had like 3,200, and despite that was lower number than in the control group, which has 3,600 hits. I think we definitely need to see what are the different methods, the counting of these hits, high intensity signal or microembol signal counting happen, and which device they use. But this is how the hits and the microembol signal really looks like. And you can see this white line resulted in the recording that when the valve crossed the arch, these hits and these microembol signal are traveling in the bloodstream. But after these passing, did not change the flow pattern. The flow pattern is continuous, so the middle cerebral artery remained open. Here's an example when this is bilateral recording. There's no embolus recorded on the right side, but on the left side. And here there's a core valve passing the arch. And after the core valve passed the arch, the signal on the left side is diminished. So this is again a nice correlation when you can see side-to-side comparison, and this is where the unilateral flow changes resulting in these microembolus signal definitely obstructing the middle cerebral artery. So in this case, we call a stroke and the acute stroke team able to retrieve the acute clot from the middle cerebral artery. That's all for me. I would like to hand over the presentation.
Great, and we'll next be hearing from Rochelle Massey. Rochelle.
Just waiting for Rico. Well, good morning and good afternoon, everyone. I'm happy to be speaking with all of you today about some of the key considerations for clinical trial design and execution for EPDs for TAVR. The first area I want to touch upon is site selection. Choosing the right sites is an essential element for not only efficient enrollment and optimized cost, but for quality patient selection as well as clinical trial compliance. The patient evaluation pathway is essential as sites need to demonstrate the processes for how patients will be identified for screening and evaluation. By working with these sites individually to outline and document the manner in which patients will be found, it will optimize the opportunity for outreach. Establishing a referral network that incorporates a multidisciplinary approach that includes a variety of specialists and specialties may prove valuable in maintaining a consistent flow of trial candidates. In terms of research staff experience and general study management, does the site have adequate resources and bandwidth to manage patient screening, enrollment, and follow-up processes? Is the research staff savvy in communicating and conveying complex trial information in a very easy way for the patients and their families to understand? It is also worthwhile to investigate what other studies they supported in the TAVR space, how they performed, and the results of any relevant regulatory agency or sponsor audits that they've been a part of. In terms of research staff experience specific to the therapy, is the site participating in other trials that would create competing priorities? If the trial protocol has an array of assessments that are nice to know but not need to know, it may create challenges for site resource allocation. Additionally, be mindful that your trial budget should account for assessments outside of standard of care. For trials assessing embolic protection devices, ensure that a consistent manner of cognitive function testing is determined and documented, as well as setting criteria for who may perform these assessments. Variability in results may actually occur depending upon who the assessor is, their experience and skills. Allowing a research coordinator versus a neurologist may net a different interpretation of results when assessing stroke. Choose your modalities carefully. Validate that sites have the proper imaging equipment. If your imaging requirements are not per standard of care, ensure that a detailed protocol and training plan is in place detailing what needs to be done and the values to be collected for data analysis. Understand from the sites if there could be potential issues with the scheduling of patients for imaging assessments, which could ultimately impact patient compliance with follow-up windows. Additionally, evaluate if there is or isn't a need for site research staff to assess the quality of imaging obtained. Unnecessary costs can be added to trials for research staff time to review and interpret imaging. And lastly, for IRB considerations, it is worthwhile to ensure that the IRB will allow pre-screening of patients. Some IRBs do have restrictions around how and when patients are reviewed for eligibility and when they can be approached regarding a clinical trial. Know at what stage the IRB will allow this patient engagement. So next, I'll move on to site training. When reviewing the protocol with the research staff at the sites, ensure that key criteria are clear from the perspective of the sponsor, especially those criteria which could exclude or include unqualified patients. Be sure on how aortic insufficiency is defined and measured. Take the time to ensure what the schedule of events that is provided is clear, detailed, and makes sense. Within the cycle of screening and treatment of a TAVR patient, does the information that the sponsor wants to collect follow the standard patient treatment sequence? By aligning assessments and data collection with the natural patient flow, you're getting the most bang for your buck. With the utilization of a novel device or in a new setting, pre-procedural training is essential. Simulation-based or hands-on training is valuable for the clinicians to get his or her hands on the device to understand how to utilize it properly and to get them truly comfortable with the technology. With the variety of EPDs currently under evaluation and the different methods for approach, it's important that the clinicians understand the technical management aspects of the device handling. Proper loading, manipulation and placement, deployment, retrieval, and conclusion of the procedure can make the difference between a successful case and one that results in patient injury. It may also be helpful to do a review of optimal and suboptimal cases. By showing the clinicians the outcomes of good and bad cases, the sharing of those previous learnings may aid in optimal results as well as the prevention of any unwanted issues going forward. Whether proctors are used within the TAVR and EPD procedure, a post-procedural post-mortem case review can be very valuable. This would include a discussion of the patient and any events or clinical symptoms they may have experienced, as well as performing a review of any imaging that was obtained pre, during, and post-procedure, highlighting what went well, maybe what did not go so well, and what would be done differently in the future, creates a dialogue between the sponsor and the ultimate end user that early on may make enough difference in EPD device success. Now moving on to patient identification and enrollment. Limit the patient search to reasonable parameters. Sites have different methods for identifying patients, and the extraction of data from a patient database may net a large number of candidates. But upon further review, some may have died or may have declined in their aortic insufficiency to a level that they would no longer be an eligible candidate for TAVR, much less to derive any benefit from an EPD. Even by limiting searches to patients seen in the past 6 months, you are targeting a more realistic population for evaluation. Secondly, patient education is essential and cannot be underestimated. With a novel or new therapy, such as EPD, patients need to understand how compliance in the trial benefits them. The EPD is not a permanent implant, therefore patients may not feel that there is value in their return visits and with following physician instructions. Simply providing a lengthy and oftentimes overwhelming informed consent does not do enough to educate a patient on why their continued participation is valued. In the context of an EPD trial, let them know that even as a control patient, they are getting specific information about their health that they would otherwise not be getting as part of standard of care treatment. Patient follow-up compliance and data collection are imperative to ensure quality data is there at the end of the study to analyze. And especially with unblinded randomized trials, it is just as important, if not more important, to ensure high compliance amongst the control arm subjects. And lastly, know your patients and remove barriers. In trials such as those for embolic protection devices, where imaging is utilized, it can be problematic as patients may not want to return for the protocol-required MRIs or imaging, which can be timely, and where they may experience claustrophobia or other symptoms. By identifying what mechanisms can be put into place to ensure that all subjects in both the investigational and control arms return for their follow-up schedules, this may affect your overall study outcomes and results. Some ideas for study visit assistance may include patient stipends for mileage, car service, taxis, or medication assistance if the IRBs will allow it. All of these are designed to help remove the burden from the patient to help with trial compliance. But be realistic and assess the patient's existing standard of care compliance. If the patient is a no-show for their standard of care visits or is not compliant with their medication, then do not pursue screening those patients. The regulatory considerations for embolic protection devices as well as other new therapies include the following. First, evaluate the need for the device. Is there an unmet need out there, or in the case of TAVR, a perceived additional risk that the EPD is designed to mitigate? Secondly, consider the trial design data capture. It's important to be cognizant when designing a trial to capture the data to show the device is needed, but also be prepared that the results may show it is not. Try to anticipate the things in the study design that could be derailers of overall success. For sponsors in the EPD for TAVR space who may be considering or are conducting a clinical trial, be certain that you want to limit your analysis to a small set of TAVR devices. With the Claret Sentinel study, the TAVR devices used were limited to the Edwards Sapien THV and XT devices, but embolic protection safety and efficacy results could vary with other TAVR products. Third, adoption is in the market. Do physicians want to use this device, and can the study provide the buy-in needed for insurance and reimbursement? For FDA, sponsors must show the device is safe. However, for everyone else, effectivity will be key. And lastly, determine if specific populations may benefit. Is there a subpopulation that may derive benefit with EPDs, such as older patients or patients with previous strokes? Perhaps when analyzed, there is greater benefit for specific, more limited populations. So what I will conclude with today is the results of the FDA Advisory Committee review. Claret Medical has submitted for de novo 510(k) approval for the Sentinel EPD for use with TAVR. Most of the panel members recommended clearance based on the safety profile and probable benefit. Unfortunately, the effectiveness endpoint was missed. However, Claret was able to demonstrate a reduction in new cerebral lesions, but no improvement on neurocognitive function. FDA does not have to take the panel's advice, but they may likely follow suit with the recommendation that was provided, and at this time, the FDA decision is still pending. It's important to note that Claret Medical has blazed a trail. With new therapies where no precedent has been set, there are challenges and always more work when a sponsor goes first. As was mentioned earlier in the webinar, the NeuroArc released proposed standardized neurological endpoints for cardiovascular trials at the end of last year. Other sponsors with EPDs for TAVR may consider utilizing these endpoints going forward. Additionally, it's key to remember that FDA is also learning along the way. Subsequent therapies may benefit from the lessons learned, and other sponsors may be able to apply those for their studies, including in areas of study design and endpoints, data collection, schedule events, and casebook design. So this concludes my presentation. I'd like to thank you all greatly for your time and attention, and I will turn it back over to Andrew.
Great. Well, thank you very much for that, Rochelle. Great presentation. Also, thank you to Dr. Garami and Dr. Lumsden for their presentations. That was fantastic. Lots of useful information there. At this point, I would like to invite our audience to continue sending in their questions and comments right now using that questions window for, of course, this is the Q&A portion of the webinar. And we've already received some questions here. So let's get started. Our first question. What other endovascular procedures have the highest instance of stroke and could benefit from EPD use?
Okay, this is Dr. Lumsden. I'll take that one. So obviously carotid stenting is an example of it's associated with strokes both from the navigation and the catheterization part and the intervention itself. Secondly, we're seeing this rapid growth of thoracic aortic stent grafts. In fact, we're involved in a clinical trial for treating type A dissections, the most common thoracic aortic emergency with devices, and strokes are also common in that situation. And so there's a variety of both current and emerging techniques that may well benefit from some sort of deflector or protection system.
All right, interesting. Thank you for that. Let's move on to another question here. What is the biggest challenge sponsors face with sites when conducting an embolic protection device trial?
Okay, well, I can take that one. This is Rochelle. So I would say that right now, getting the physician buy-in to the efficacy of EPDs is the greatest challenge. Most physicians want to practice evidence-based medicine, and without the concrete efficacy, I believe it creates a barrier to having that confidence to introduce a new device into a procedure that already has its own inherent risks. Once other EPD studies are conducted and there's results that show efficacy, I believe that will give the physicians the confidence that they need to utilize the device in their procedures and have that kind of contagious enthusiasm with their patients and supporting staff as well.
Right. Okay. Thank you for that as well. Another question, I believe, Dr. Grammy, this was asked during your presentation. Are baseline flare analysis enough?
I would like to say no. The DWI is the only way you can see any new acute event. So if you do a baseline and say DWI will tell you that within 7 days, is there any new acute event? The flare going to give you the last 80 years, all the accumulative stroke happened in that brain. So that's definitely my point of number one. Number two is that that baseline really needs to be done a day before or even the morning of that procedure. So it's very important that we definitely don't do like 7 days before the procedure because we're not going to have useful information. And the second reason we are using the DWI, for example, to really track any of those new lesions, that we're able to generate even a substruction image. So for example, day 2 image would be overlaid with the baseline DWI image, and this is how we can see a difference. And also I would like to comment that this is one of the reasons why the time is very important and the days. So we need to document where this baseline DWI is done and the post-procedure DWI is done. So the start and end time of each of the procedure and the MRI scan is a very important time point for any of these trials.
Okay, thank you for that. Another question here, can TCD differentiate clots from air embolization?
Right now, this is the most difficult we're trying to solve as a big question for us. We can tell that if something else traveling in the bloodstream is not a red blood cell, but we cannot tell this is clot or emboli. But what we can solve, making timestamp notes in the operating room to see what is the step of the procedure, is there any catheter movement, or is there any contrast injection, which has me participating some air embolization from, or the other part of the procedure. We tried with dual frequency TCD 2 and 2.5 megahertz to differentiate them, but the large air emboli or large air bubble looks like a clot on the sound signature. But we are working on it to have more accurate counting and also answer the this question in the future. Thank you.
All right, thank you for that, Dr. Grammy. Another question here, perhaps for Dr. Lumsden. Do you think there is a role for MRI and TCD during standard open heart surgery? And also, why has TCD not been more widely utilized? Your thoughts on that?
Thank you. It's a great question. Again, when I read the NeuroArt classification system, my kind of eyes were getting bigger all the time because obviously the focus at the moment is TAVR. But if you start really implying that these MRI-related lesions are strokes, and we're going to end up having to go back and look at a whole variety of previously established interventions. So, for example, after open heart surgery, people also describe pump head, confusional states. Patient's just not right for a period of time. And so big question is this microembolization that we're dealing with? Is it hypoperfusion, which is another thing we could actually tell using TCD. So I think this is going to be lead to a lot of reevaluation of what are currently fairly standard treatments to see whether or not this is truly microembolization. Now, the question about TCD, why is it not being used more widely? It's a remarkably, in my opinion, remarkably effective way of looking at blood flow in the brain, direction of blood flow, the velocity of the blood flow, change from baseline when we're doing, for example, carotid endarterectomy and looking at embolization. And the technology is actually remarkably simple. These are not expensive machines to buy. The challenge has really been on the kind of the user end in terms of doing the interpretation of it, and that has been a challenge. And but we've actually kind of been developing ways of monitoring these remotely, so that you don't necessarily have to have somebody like Dr. Grammy on the staff. You probably kind of like EEG monitoring is often done during carotid endarterectomy. It's just a matter of putting it on, and I think increasingly these will be able to be monitored remotely. So some of those challenges which have limited it in the past, I think, are now being addressed, and it's with new technology and remote sensing.
All right, interesting. Thank you for that. Another question here for Dr. Grami. Can you say again why you'd rather use a 3T than a 1.5T MRI?
Yes, so because of during the TAVR procedure, these lesions are extremely small, and it's difficult to detect them because the slice thickness, we would lose the partial volume effect, and we want to have a better sensitivity to detect these. 3T is giving us higher resolution to really go after these tiny lesions. Benefit also that we also have a faster scanner, so we are shortening our scanning time, probably make it more reasonable to do the fast scan before a procedure, right even the morning of that procedure. And also more and more new sequences are coming that these are the cutting edge, and I think the high-end 3T really needed for evaluating these small lesions.
All right, excellent. Thanks for that as well. Dr. Lumsden, could you elaborate for us on the factors that affect the porosity of a device and its ability to capture or deflect debris?
Yeah, thanks. So again, one of the things we've learned from carotid stenting is just having a device or using a device doesn't necessarily mean it's completely effective. And so the things that are going to have to be defined is what is the porosity, what is the material that is made up of, and almost all of them have some sort of hoop or seating system. And so that they may or may not seat. There may be flow which is going around them. We know also that as reduced porosity, you capture more particles, but at the same time, the thrombogenicity may actually go up. And so what you clearly don't want is a situation where you put an embolization protection device which captures particles but generates its own thrombus. So there are a whole series of biomaterial considerations and seal zone considerations that are going to have to be evaluated. And that's really what we're going to be able to try and figure out. And that's one of the reasons that I am a complete advocate of real-time monitoring. If not in every patient, but at least in the phase 1 of these trials. So we try to refine exactly how these devices are performing and being able to tell real time whether they're truly effective in preventing embolization.
Okay, thank you for that. We'll try to squeeze in as many questions as we can here. Next question, what is the clinical significance of microembolic signal in asymptomatic MCA or carotid stenosis? Dr. Grammy, maybe you have an answer there for us.
So this is really interesting because this is a pre-procedure screening. TCD monitoring can evaluate if we have a embolic source in the middle cerebral artery or in the carotid artery without any symptoms. So when you have these microembolic signals, in our previous studies, we showed that if within a year all those asymptomatic patients have a 16 times higher stroke rate. So I think the data is already there, and I think the microembolic signal is a pretty good predictor that if you are asymptomatic today, you may not going to be asymptomatic tomorrow. Thank you.
Okay. All right, thanks for that as well. Maybe two more questions here. Should centers that lack TCD capability be excluded as a potential study site in clinical research studies of embolic protection for TAVR?
I would say I would definitely exclude them in the phase 1, as Dr. Lumsden just mentioned, for the reason that during the learning time, we really would like to get as much information and much data as possible. And I think comparing the TCD data to an MRI and to a cognitive function, also the clinical outcome is quite important. And I think TCD is a very important part of this puzzle.
Right. Okay. Well, thank you very much for that, Dr. Grammy. Also, thank you to Dr. Lumsden and Rochelle Massey for all of your answers. We have run out of time today. We have reached the end of the question and answer portion of this webinar. If we weren't able to attend to your questions today, however, the team at Novella Clinical may try to follow up with you after the webinar. And for more information, I encourage you to visit the websites showing on your screen right now. That's medicalmetrics.com and novellaclinical.com/MDD. And thank you everyone for participating in today's conference. You'll be receiving a follow-up email from X-Talks with access to the recorded archive for this event. And a survey window will be popping up on your screen shortly, and your participation is appreciated as it'll help us improve our further webinars. Now, I'm just about to send you a link in your chat box. You can say thanks to our speakers for the presentation today, so I encourage you to do so as well. Thank you very much, Rochelle Massey, Dr. Zsolt Garami, and Dr. Alan Lumsden. We hope that you found this conference informative today. Have a great day, everyone.
Okay. Thanks.